Hello again, I'm Reba Zimmerle.
And I'm Torline, and our presentation is called The Identification of PLA2 Properties in Snake Venom Based on DNA Sequences.
PLA2s are phospholipase A2s, which cleave the phospholipid bilayer found in cell membranes. They are found in a variety of animals, including mammals. In mammals, they aid in fertilization, smooth muscle contraction, and cell proliferation or cell growth.
So while PLA2s are very helpful in mammals, they can have a variety of effects when they're found in snake venom. This is because the PLA2s in snake venom have what is called a pharmacological site. This pharmacological site is separate from the catalytic site, which performs the enzymatic activity of breaking apart phospholipid bilayers. This pharmacological site targets membrane proteins that are found in various tissues and organs throughout the body. This means that when the PLA2s enter the body, they can attach themselves to tissues and organs that are around your body to exert their toxic effects.
There are two main kinds of toxicity in snake venom, one of which is neurotoxicity. Neurotoxicity is the inhibition of the firing of neurons, leading to paralysis and possibly death. Two examples of neurotoxic proteins found in snake venom are three-finger toxins found primarily in the Elapidae, and neurotoxic PLA2s generally found in Colubridae. An example of a highly neurotoxic snake is the black mamba.
So of the neurotoxic PLA2s, there was one that was very important for our research, and that was crotoxin. Crotoxin was first identified in Crotalus durissus terrificus, which is a South American rattlesnake. And it is the most classified and best studied of the neurotoxic PLA2s. Now, crotoxin is actually made up of two subunits, crotoxin A and crotoxin B. On its own, crotoxin A is completely non-neurotoxic, and crotoxin B is only slightly neurotoxic. However, when the two subunits interact to form a protein complex, they become greatly more neurotoxic. This is because crotoxin A inhibits the non-specific binding of crotoxin B with membrane proteins on muscles, which means that the protein complex can only bind to neuron proteins and can only exert its toxic effects there. This can make the neurotoxic effects of crotoxin B up to 30 times more powerful.
Another kind of toxicity in snake venom is myotoxicity, which affects muscles and tissues. This is the classic snake bite effect that you think of in reference to rattlesnakes, really red, swollen, bloody.
So some of the symptoms that are associated with myotoxicity are blood loss, swelling of tissues and organs, the discoloration of skin, and necrosis, which is the death of living cells. PLA2s specifically cause necrosis by breaking apart the cell membranes that hold cells together. So snake venom is very important to snakes, obviously, because it helps them to kill and digest their prey.
PLA2s are very important in snake venom because they are found in pretty much all snake venom and have such a wide variety of effects. Part of the reason for this variation is accelerated evolution. Some things you should know. One, exons are the parts of our DNA that code for proteins and other characteristics. There are genes. Two, introns are the part of our DNA in between the exons and don't code for anything. Three, introns generally mutate more than exons.
However, this is not the case in snake venom. Studies have shown that in snake venom, the exons actually mutate faster than the introns, causing more variation in the proteins found in snake venom. So in our study, we decided to sequence the PLA2s found in two snakes, the Mexican West Coast rattlesnake, or Crotalus basiliscus, and the Yucatan neotropical rattlesnake, or Crotalus thymus zobcon. We also compared the sequences that we found to known sequences, which we got off of the National Center for Biotechnology Information website. Our hypothesis for this study was that by comparing our sequences to that of known sequences, we could determine the functions of our PLA2s.
So Crotalus basiliscus is a very large rattlesnake. They have very large glands and can yield a lot of venom. However, their venom has not been studied much. A previous study did recover a partial amino acid transcript from a protein, but beyond that, not much is known.
Like Crotalus basiliscus, Crotalus sinus dogcon has very little that is known about its venom. It's known that this venom can cause neurotoxic effects, but it has never been classified or sequenced. Here we have a map showing the ranges of our two snakes. As you can see, Crotalus basiliscus is found along the western coast of Mexico, and Crotalus sinus dogcon is found in the Yucatan Peninsula. It's worth noting that these ranges don't overlap.
Now on to the mechanics of our project. The first step was RNA extraction. The samples that we used did not just have RNA, so we had to purify it from the rest of the compound. RNA is a very unstable molecule, so we had to be very careful in this step because RNases, which are enzymes that break down RNA, are found everywhere, on your hands, on tables, in the air. But once we did successfully extract the RNA, we measured the concentration using a nanograph, which uses wavelengths to do so. This verified that the extraction was successful.
After that, we had to convert our RNA into cDNA, or complementary DNA. Complementary DNA has its name because it is the DNA that only includes exons, so it's only the DNA that codes for proteins. We also added primers to our DNA. These primers use conserved regions that are found in all PLA2s to make sure that we would only get the DNA for PLA2s and not other proteins.
After cDNA synthesis, we did have DNA, but it was a very low quantity of DNA. We had to amplify this amount, and we did so using PCR, polymerase chain reaction, in which our sample and polymerase, which is the enzyme that makes DNA, are cycled through several different temperatures, which are all well above body temperature, to stimulate polymerase into making many, many copies of DNA.
We then put our samples into a gel electrophoresis, which uses the slight negative charge of DNA to separate it based on its size. PLA2 transcripts have a known size, and so they form bands in a specific size range, and we were able to cut these bands out and isolate them from the rest of the DNA. We then had to purify the DNA from the gel, and we used spin columns to do this.
Once we had the pure DNA, we needed to amplify the amount again. We did not use PCR because polymerase tends to make mistakes in the process. Instead, we inserted our sequence into a plasmid, which is a ring of DNA that E. coli and other bacteria naturally take up from their environment. You can see here in this picture one of our control plates of E. coli, and this proves that the E. coli successfully took up our plasmid. After that, we purified our plasmid by picking colonies from the plates, growing them separately, and then purifying the plasmid from the colony.
We then sent our DNA sequences to Arizona State University, where they were sequenced using a method called Sanger sequencing.
Once we got our sequences back, we analyzed them using a program called Cronus Lite, making sure that all of our sequences were of a Fred quality score of above 30. We then put our sequences into BLAST, which is the basic local alignment search tool. This compared them to other known PLA2s. We did this so that we could confirm that we definitely had PLA2 sequences and to figure out what type of PLA2 they most related. We then converted our DNA sequences into amino acid sequences. Once we had our amino acid sequences, we aligned them in FASTA format with several similar amino acid sequences. It should be noted that we received transcripts from our samples that included acidic PLA2s, but we focused on the crotoxin transcripts instead. In crotoxin B, we looked at a specific location, which is asparagine at location 6, which indicates neurotoxicity.
We also ran a protein gel with our venoms, and we did this so that we could analyze the protein content of the venom and make sure that they definitely had PLA2s in them.
Now for what we found out. As you can see in this picture, both of our snakes produced a band of DNA around 600 base pairs long, which is the expected length for PLA2s.
In our protein gel, both of our snakes produced bands that were in the 13 to 15 kilodalton range, which is also the expected size range for PLA2s. This is our sequence alignment, which you can't really see now, but we did two separate sequence alignments, one for crotoxin A and sequences that were similar to that, and one for sequences that were similar to crotoxin B. In this sequence alignment, dots are used to indicate amino acids which are similar to the ones at the top.
Some other things to note is that the bolded sequence at the beginning indicates the signal peptide, which tells the cell to excrete the protein, and there are percent identities after the sequence, which is how similar the line is to the top line.
So for our Crotalus Zobcon sequences, we found a sequence in Zobcon which was very, very similar to a sequence of Crotoxin A from Crotalus terrificus, and a sequence which was identical to Crotoxin B in Crotalus terrificus. Because our protein gel indicated that we had PLA2s in the venom, this means that we likely have, that Crotalus terrificus likely has PLA2s which have the same functions as Crotoxin and therefore has highly neurotoxic venom. Here in our sequence alignment for Crotoxin A, you can actually see that Zobcon and terrificus had a 99% identity. They actually only differed at one amino acid, which is here, but where they differed, Zobcon is actually identical to another PLA2, which has the same function as Crotoxin, indicating that this difference likely didn't affect the function of the protein. In Crotoxin B, Zobcon had a 100% identity to terrificus, meaning that it likely had the same function.
Our results for Crotalus basiliscus were just as encouraging. The sequence that we recovered for Crotoxin B matched the partial sequence that the previous study recovered perfectly. In our alignment, you can see that it has a 100% identity with terrificus. So it is neurotoxic. However, we did not recover a Crotoxin A subunit from basiliscus, indicating that either it's only slightly neurotoxic or we just failed to recover that subunit in our experiment this time. Again, our match. The similarities between these Crotoxin subunits might be attributed to hybridization. However, this is not possible because they live in entirely different regions. And Crotalus terrificus also lives in South America, so they wouldn't be able to hybridize. So therefore, the similarity is probably due to a common evolutionary ancestry or to a very similar habitat forcing a similar protein development. The next step in this experiment would be to purify and sequence the proteins from the snakes and to verify that the transcripts produced from the protein match the same transcripts that were produced from the RNA. If they match, the next step would be to test and make sure that the proteins had the expected effect.
If they did have the expected effect, this would indicate that we could identify the functions of PLA2s without testing them on mice. This would be pretty important because sometimes venom can have a different effect on mice than it does on humans. So having another way to determine the function could make it more accurate. Along with that, because we got the DNA sequences for our PLA2s, we could use that to do gene modification and change some of their amino acid sequences, and that would help us to learn more about the structure-function relationship in PLA2s.
And by learning about the structure-function relationship, we can learn why PLA2s are able to target specific cells so well, leading to possible cancer treatments, for example. So here are our citations.
We also have quite a few people we'd like to say thank you to. I'd like to say thank you to my scholarship and research sponsor, Excel Energy.
I would like to say thank you to the Torline Family Foundation for funding my scholarship here, and to the Kinder Morgan Foundation for funding my research.
We'd like to give a very special thanks to our research mentor, Kathy Modal, for helping us through every single step of our research. We absolutely could not have done this without her.
We'd also like to thank Dr. Stephen McAfee for the use of his lab, a chance to get in on his research, and for the brief time that we got to interact with him, his amazing expertise.
We'd like to thank Amy Dunn, our research advisor, for reading through all of our papers and watching our practice presentation and making sure everything was as good as it possibly could be.
And then, of course, we'd like to thank everyone else in FSI for being amazing friends, and to our teachers and everyone else we interacted with for just the amazing experience of learning so much.
We'd also like to thank our families for supporting us.